And now we're going to look at the reads quality control. One thing I forgot to tell you is that in RStudio you can run the code by clicking this button, this green triangle button. So you don't need to copy paste your code to the console. You can run it from the file itself. Yeah, I'm sorry, I forgot to mention that before. So let's go through the reads chapter and see whether it's different from UMI counts. So first of all, if we look at the expression metrics, we immediately see that the number of counts here is much higher than before when we looked at UMIs, right? So this is not surprising because with reads we always have amplification, such as PCR amplification, and you always have much larger numbers here in the metrics. So annotation table is the same. Then we create a single cell experiment object. Again, removing the genes which are always 0. Defining ARCCs and mitochondrial genes the same way. Running calculate QC metrics function. And first we look at the distribution of total counts. So this plot looks very different from what we saw before. First of all, again, the scale here is much larger. So the number of total counts in the read matrix is much higher. And also the distribution looks a bit different. So it looks kind of bimodal. So we have some cells here, one peak, and then there is another peak here. So again, this is very subjective. So we decided to remove all the cells in the second peak and only leave these cells, right? But here you can see already the difference between UMI and reads, and also, so this may be created by amplification bias. It can be related to amplification. It can be something else. But if you work with reads, you have to understand that your values are very different from molecules. So molecules are usually much cleaner, and you can get more sense from looking at these figures. With reads, it's a bit more complicated. So if we remove these cells, this filter removes about 180 cells in total. Then we look at the number of expressed genes. A number of expressed genes, I think, looks more or less the same. So again, we remove genes, remove cells which have less than 7,000 genes expressed. I think it's actually very similar to the figure we saw before, which means that the position, the indices of the expression values are the same. So you have probably the same genes expressed in the same cells, but the values of expression are much higher, right? Yes? So the question is, do we analyze UMI first and then reads? I think usually you have only one of them. So you either have UMI or reads. So your analysis will depend on what the experimental protocol is. So it's just in this paper they did both, and we can actually compare and see the differences. Yes. So the gene filter removes about 116 genes. Then we look at the mitochondrial genes, and now we see that percentage of counts in mitochondrial genes is even higher than we had before. But there is no, here again, there is no strong batch effect. So everything is kind of mixed. So we would cut something, maybe 20%. define those cells as outliers and keep the rest. If we look at the ERCCs, so again, the counts are much higher. So some cells have 60% of counts as ERCCs count. But the batch effect is still here. So this is a good indication that the batch effect that we saw before is real. So we see it in reads as well, even though the amplitude is a bit lower. In UMIs, it was much cleaner and stronger. So actually, now because ERCC's counts are so high, in addition to removing the batch, we also remove cells with more than 25% of ERCC's counts. So here we have an additional filter on ERCCs. So this removes about 103 cells. For mitochondrial, we remove everything more than 30%. This removes 18 cells. So then if we combine all 4 filters together, we remove about 258 cells, which is more than in the UMIs. You can compare the cells, like did we remove the same cells or not the same cells? You can do that as an exercise. We don't have it in the course, but that's a good idea to check whether the overlap between those outliers is significant. Now if we run the Pca automatic identification of outliers, I think it looks better on this plot. So now we have a definite outliers on this side, and Skater defined them quite nicely. This filter removes, is it false true? Okay, so this filter removes 108 cells. So if we look at on the overlap of the filters, again, our manual filter includes all these 108 cells that Skater identified, but additionally we have 150 extra cells. So our filter is much stronger, and usually the more you remove, the better. So don't leave anything suspicious. Yeah, we can also look at the top 50 expressed genes. Again, we have some ERCCs here. And if we had gene names, probably we could guess some biological meaning of this data set. But unfortunately, we only have ensemble IDs. The gene filter, if we use the same gene filter as before, so we remove genes which are expressed in less than 2 cells. This gene filter removes about 2,600 genes. Yeah, so if we apply both filters, this is the data set that we get at the end. Again, we want to log transform the counts and write it to log counts underscore raw SA. And then we want to save the file into this one. And I think that's mostly it. Do you have any questions? Yeah. Could you repeat it again? I didn't understand. So you mean some specific ERCC in the data set? This one? So the question is, we have a spiking which has a very variable expression across cells. So do we ignore it when we normalize by spikings? I think when you normalize by spikings, you don't ignore any of the spikings. So you need to make sure that you have good spikings in the first place. Yes, so ideally you would like it, well, sometimes, I mean, depends on your experimental condition. So it should not be the same in every cell. I mean, the whole point is, the assumption is that the concentration should be the same. That's why you normalize. But if it scales so much, maybe it's not a good spiking yet. Maybe you're right.
And also, it's a sort of it's well known that for mRNAs, the variability is proportional to the mean expression level. So highly expressed species will have higher variance. That's just what you expect to see. So that's why this happens as well.
So the question is, is it the reason for that because RCCs are shorter than mRNAs? I don't think so, because RCCs are bacterial RNAs, so it doesn't really matter whether they're short or not. Shouldn't matter, at least. But I'm not sure. Okay, any more questions? Okay, so now we move on to the next chapter, which is about data visualization. So far we have done quality control of our data set. So we removed all the bad cells, all the bad genes, all the bad cells. Now we want to look at the data in more general way. So the first thing we can do is a Pca plot. I hope everyone is familiar with the Pca plot. Pca is abbreviation of the principal component analysis. And in a very simple way, what it does is it projects your data into a plane in which the variance of the data is the highest. And then it defines it as the first principal component. Then it finds a orthogonal plane to this with the highest variance, defines that one as a second principal component, and so on. So at the end, what you have is usually you plot PC1, principal component 1 against principal component 2, and it gives you some idea of what's the structure of your data. So we can first look at the raw data without log transformation. So we have the counts assay in the single cell experiment object, and Skater allows you to plot Pca plot very easily using this function. And we say we want to use counts assay as a data. We want to color by batch. We want to size by total features. Total features is the number of expressed genes in the cell. And we want to shape by individual. So all these things actually come from ggplot function. So you can specify different aesthetics. If you are familiar with ggplot, Skater integrates ggplot, so you can use your own aesthetics here. So if you plotted the raw data, interestingly, we can see that the batch that we found using ERCCs is visible here as well. So it was good that we confirmed that this batch is bad by looking at ERCCs. But in general, we can see that we have 76% of variance on the first principal component and only 4% on the second principal component, which means the batch effect is much smaller compared to to this effect. And this effect, if you look closer, you will see that here you have small size of points, and then it increases if you go to the right, which means that the variance on the first principal component is mostly described by the by the number of total features in your data set. So the size, we define the size as a total feature in the cell. So, and this is very common for single cell data sets. Usually the first principal component always correlates with the number of total features in your cells. Oh, sorry, I'm speaking Russian now. Yes, so features are your genes, yes, yeah. So before plotting Pca for single cell RNA-seq data, you have to normalize it by the library size. And then this variance will shrink a bit. So this first principal component is an artifact of the number of expressed genes in your cells. Now, if we look at the log-scaled counts, the ones that we defined at the end of the previous chapter, right? So this one is already a bit cleaner. So first of all, the variance here is a bit smaller. I think we still can see the size, the total features effect on the first principal component. But now we can also see separation of the patients, of the individuals. So here we have individual 1, individual 2, and individual 3. So there is a bit of separation between individuals. So that it gives you an idea that log transformation is usually a good thing to do. So it allows you to kind of zoom the data and see the better overview of it. I think I need some water. Sorry, just a second. So I think the log transform data looks already better on the Pca plot. And I think that's what we say here. Log transformation is beneficial for our data. It reduces the variance on the first principal component and separates biological effects. However, this is important. Keep in mind that simple log transformation is not enough to account for different technical factors between the cells. So this is not a proper normalization. So we will cover normalization in the next chapters, but keep in mind that simple log transformation doesn't solve all your problems. So it's not enough. It's not a normalization step. So the plots that I showed you before, that was on the original data set with all the cells included. So we didn't exclude the outlier cells and genes. Now we're using a different object here, UMI-QC, which is already filtered object. So we removed all the outlier cells and outlier genes. And again, we look at the log-scaled counts. And now we can see that it is even better. So again, the variance decreased to 17% on the first principal component. So there is not that strong effect of the total features. However, we only have one individual separated from the other two. The other two are more mixed together. Also keep in mind that plot PCA function in Skater by default uses the top 500 most variable genes. So you can control this in the function by changing this parameter. So I think this is an exercise. So you can control the number of genes that you want to use for your PCA by ntop parameter in the plot PCA function. And as an exercise, we ask you to run the PCA, plot the PCA plot for all the genes and for only 50 genes. Yes?
Do you know how it determines gene variability? Like, how does it rank?
So the question is, how does Skater determine variable genes? I think Davis can answer.
Variance on whatever scale that your expression values are.
Variance over mean.
Just the variance. Variance.
Okay, so it's just defined by the variance.
Yeah, it's and by default, it will scale of features. So generally, you just want to do this on log scale counts because that's closer to the sort of normal scale that PCA says. As an illustrative purposes, for illustrative purposes, we've looked at like doing PCA on raw counts, but you would basically never want to do that in practice. Yeah, so normally we do this on log scale, normalized counts. That's sort of the most effective use of PCA, I think. And so in terms of a single cell experiment object, by default, the plot PCA function will look for that log counts element, and it will use that by default unless you tell it to use something else. And one final thing, log counts is in Skater is synonymous with the function express or exprs. So we incorporated that for backwards compatibility with SCE set objects and stuff. But you can access those log counts either with the log counts function or with exprs, and they'll give the same stuff.
So if you run the previous function here, plot PCA, but you need to add ntop parameter at the end of it. And run it for 50, and this number is probably wrong. So do you remember what was the number of genes in the UMI dataset after filtering bad genes? Can you tell me? Yeah, so I think we didn't change this. This number is from the previous time we ran the course. So you need to look at what this number is and see how different your PCA plot will be if you use only 50 genes or if you use all the genes. So far we always used 500 by default. So I hope you can run it easily. So, or you can use the RMD file again in RStudio for your convenience. So if we use all the genes, I think what we see is that the feature size, total feature effect becomes even stronger, even though the variance here is small, but we can see that we have all the small dots here, and then it increases if we go to the right. But also we see that all the features, when we use all the features, we separate this individual again, the third one, from the other two. So there is some biology here as well. If we only use 50 genes, so this is very interesting now. So we see the effect of total features So in every cloud on this plot, we can see that you have smaller points on the right and bigger points on the left, but also we separate batches from each other. So this batch is now from this individual. So we have two batches together, and one batch is separated. In these individuals, they're kind of together. The third one we removed by quality control. And in this individual, all the batches are together, right? So here we can see a bit more biology instead of total feature effect, which is not surprising because we use less genes. So the number of total features in those genes, the noise and the variance in the total features is less because we have less genes. So the next plot that we can use to visualize our data is a t-SNE plot. It's become very popular with single-cell RNA-seq data especially. So if you go to any single-cell RNA-seq conference, you will only see t-SNE plots. Every talk has a t-SNE plot. However, you have to be very careful with t-SNE plots. So t-SNE plots are very good for visualizing your data, but they shouldn't be used for clustering your data. Because with the t-SNE plot, you can, by changing the parameters, you can make it as beautiful as you want. And it can be very different depending on the parameters. So it's not good for clustering, but it can be very good for visualization. And also, it's stochastic. So if you run it multiple times with different random seeds, you will get different results. Whereas PCA is deterministic, so PCA will always be the same. So Skater has a plot t-SNE function. It's basically the same thing. So we just change plot PCA to plot t-SNE and use the same values, except that we fix the random seed, because we want to see the same results. And we also define the perplexity parameter. Perplexity is a parameter which defines, because t-SNE is, I should have told some more information about t-SNE, right? So t-SNE is the abbreviation for distributed stochastic neighbor embedding. So what happens here, you kind of embed your data into a 2D space, and perplexity parameter defines the density of your points in this space. So if you change the perplexity parameter, you can make this plot look very different. So here we fix, I think this one is there is a default parameter, perplexity parameter. I don't remember how it's defined.
It's usually about 20% of the total number of cells.
Okay, so the default perplexity parameter is 20% of the total number of cells, which is probably this number. And if we,
Is the R in the rank node? Is it like the chance of going back to the same node that you started from?
It's something sort of like, it's like the number of expected neighbors for each given cell.
okay, so yeah, so perplexity, as I said, it's related to the density of points on your plot. So as David says, this is a number of expected neighbors for each cell. So by changing this, you can change the distribution, density distribution of the points on the plot. And as you can see with this data set, at least with this perplexity value, the plot looks really funny, I think. You can't say anything about variance on this plot, so it's not like PCA. You can only see that these batches, for example, for this individual are separated a bit. These batches are also separated. This individual is also, like, we can see that there are 3 individuals. The batches inside each individual are separated, but we can't say anything else about it. Now, if we change and this was with the row counts, so we use the we use the original data set with all the outlier cells included. Now if we remove the outlier cells and outlier genes, it actually becomes cleaner. So we have this individual here with some cells, they appeared on a bit far away from the rest. This individual here, but some cells of it are on the other side. So maybe these cells are bad, maybe not, but we didn't see that effect on Pca plots, right? Now we see it on t-SNE plot. Yeah.
But in this case, after the quality control, you removed one of the replicates. The data photograph.
After quality control, we remove one of the batches, yes.
Yes.
Yeah. So, as an exercise, we want you to run t-SNE with the perplexity parameter of 10 and 200 and see how the plots are different if you change the perplexity parameter. Yeah, so I'm not sure how it's related to density though. So if we change the perplexity to 10 here, right, we see more or less the same picture. It's really hard to say whether the density of points decreased or increased. If we change it to 200, here probably we can see that the points are more separated from each other, right? So I think when you increase the perplexity parameter, you separate points more. If you decrease it, the points become closer to each other. Okay, do you have any questions about t-SNE and Pca plots? Yeah. Random seed is just any number. You can choose. Yes. So the point of random seed is that if you run it on your computer with the same random seed, you should see the same plot, right? If you change it, you will see a different plot. So we set the random seed to this value. You can use just one. Random seed equals 1.
For example, I don't say the random seed, but I just run it and it looks good, and I want to have that plot again. Can I find the random seed?
You can't find the random seed for what you did before. Yes.
You don't define it, it's usually done on the time.
Yes.
That's the default in most software packages. It's just like the current, whatever the current time is, it uses that as a
Yes.
seed.
So when you run it, it will be connected to the time of running. When you run it afterwards, 2 minutes later, it will be different anyway, and you won't be able to figure out what was the random seed at that time. So when you run it, it's
random numbers in software are not actually random. They're pseudo-random numbers, which means they come in actually a defined way, which is very difficult to predict if you don't know where they're starting from. So that's why they look like random numbers, but that allows us to do things like fix a specific point in the sequence of random numbers, is defining a seed, and that makes things that would otherwise appear random become reproducible.
yeah. So that's
So when you write the random seed, you see a name that run without conditioning.
when we define the random seed, we actually say instead of using your machine random seed at the moment, use this random seed. So when you run it on your machine next time on a different system, it will use the same random seed, so you'll see the same picture. Yeah.
Silly question, maybe. Like, what's the function of the random seed? Like, because if you don't set it by, like, if you do set it by default, then it's great. But if you don't, then you can never reproduce your own plot.
Yes, because it's stochastic. It's stochastic. It's like throwing dice. If you want to reproduce throwing a dice, you need to record every movement on your trajectory, right? If you don't, then next time you throw it, you will get different result.
And why do you need that
We need that, so when you run on your machine, you will see the same plot.
No, like in general, like why is it in there?
Why is it stochastic? Why t-SNE is stochastic?
Yeah.
That's how it was developed. It's based on the stochastic theory, right? So you can't control this. It's basically.
I think in general, people develop stochastic methods because they're sort of faster. It allows you to speed up things, and also if it's a case that whatever you're trying to solve for is analytically intractable, you can instead basically use Monte Carlo, as it's called in sort of fancy speak, to approximate the solution. I'm not expert on t-SNE, so I'm not exactly sure why it's stochastic, but that's the general why you want to do that.
I think usually when you can't find the exact solution, you approximate using stochastic algorithms. And the disadvantage is that you get different results when you run it. So to be able to reproduce it, you need to fix the random seed in the beginning, and then it will do the same calculations based on this random seed. Sorry?
They will be different, but not that different. They will still be very similar, right? It's not that.
Yeah, so they will be different. Yes.
You will definitely get more or less the same picture otherwise.
Quite often it's just rotated by 90 degrees or 80 degrees, but if you're quite often it's just rotated by 90 degrees or 180 degrees, but if you're trying to add annotations or anything to your plot after you've plotted the points, that's going to be a big problem for you.
Yeah, so if you are preparing your data for publication, it's better to fix the random seed so that when you rerun it in a couple of months' time, you will see the same picture. It will not change, right?
And Lawrence van der Marten, who's one of the two people who developed t-SNE, explicitly said, it's a way of making nice-looking plots of high-dimensional data. And so his advice is, run it as many times as you like until you get a picture that you like the look of. So, he doesn't take it too seriously, and the big lesson is we shouldn't over-interpret these plots. They're one way of getting a really high-dimensional data set into 2 dimensions so that we can look at it on a page. And it has to do a lot of things under the hood to make that possible. And so one of the traps for interpreting t-SNE plots is that unlike a PCA, where we've got 2 well-defined sort of length dimensions, the first principal component and the second principal component, t-SNE does not have this property. So the length scales are different at different regions of this 2-dimensional map. And it does that so it can try to capture what it understands as the high-dimensional structure of the data in 2 dimensions. Because the big problem for all of these approaches is that there's a lot more space in 10,000 or 15,000 dimensions than there is in the 2 dimensions that we want to look at. And so this is this idea of this known as the crowding problem. How do you faithfully represent the distances between points in a very high-dimensional space when you only have 2-dimensional space to work with? And so t-SNE tries to do this in various ways, and one of the the trade-offs is that it's going to sort of zoom in on elements of the structure and display them in parts of the 2-dimensional map so that we can't interpret distances in the top left corner in the same way as distances in the bottom right corner or vice versa. So
it's just the visual inspection of your data, nothing else, I would say. Also, we provide a link here to the website where you can see how t-SNE changes if you change some parameters. So for example, here we have 2 data sets. This is the original data, right? You have 2 clusters separated. So, and you can see how it changes when you change the perplexity parameter. And also depending on how many steps you run the t-SNE for, because it's stochastic, it has some number of steps to run. I think by default in Skater, I don't know the number, but you can get anything you want with t-SNE. That's the message from this this website. Yeah. If you run t-SNE on 2 samples, 2 experiments, and you want to see the differences between them? So the question is whether it's a good idea to use t-SNE on 2 different experiments to find out the similarities between the experiments. I think this is a bad idea, as Davis just mentioned. On a t-SNE plot, the distances do not correspond to real distances between the cells. So it's better to use Pca plot for these purposes. And we will discuss that later, actually. Yeah. Yep.
Usually, how do you decide what's going to be your perfect recipe? Is it the protocol that you use to determine your single cell data, or is it like it depends on the biology of the tissue that you're trying to observe?
So the question is, how do we define perplexity? I think there is no recommendation here. So it's as we say, you want to show something with t-SNE plot. So you find the perplexity value which shows your message, and that's it. I know it's funny, but it's in the papers. It's sometimes you can do separate clustering of the cells separately from t-SNE, and then visualize your clustering using t-SNE. Actually makes sense, because sometimes t-SNE can separate exactly the clusters that you identified, whereas Pca would not do that. So for visualizing your clustering, it's a good way to do it. But you have to make sure that your clustering is not based on t-SNE dimensionality reduction, right?
So is the perplexity somehow correlated to the restart coefficient in random walk with restart? Because normally they define coefficient of restart.
I don't know. So the question is whether perplexity is correlated with the random walk, start of the restart coefficient of random walk. I don't know. Do you know?
I know what you're describing is more similar to a diffusion map sort of approach, which is also used in various ways in single cell data. So no, a t-SNE plot, in my understanding, it's not interpretable in exactly that way. Even in the original publications, they're kind of a bit vague about exactly how to interpret it, and so the closest I can get is that, loosely speaking, it sort of defines the number of expected neighbours of a given cell, and so a lower value of the complexity, so expecting fewer neighbours per cell, will generally correspond to tighter groupings of cells, and larger values expecting more neighbours per cell gets those more sort of strips of cells in the t-SNE space, because it's sort of trying to account for that. And the crux of t-SNE is that it's trying to use conditional probability distributions as ways of defining distances, and so it takes the conditional probabilities of sort of 2 cells being in the same space in the high dimension, and then uses a t distribution in the low dimensional space to try and define, get appropriate distances from that conditional distribution in that low space. But yeah, it's a pretty involved technique, so I think if you really want the details, go to the publications, because they describe it better than I can.
Any more questions? Good. So now we would like you to do the same kind of plots for reads. So I've done this for UMI counts. Now you can do it for reads. Again, you can use RStudio and run everything there, which is easier. And we'll give you 5 minutes and then continue.